Okay, thank you. Can you hear me with this microphone? Because I might move around. Chad did a wonderful introduction on CRISPR, so I'm not going to give you any background. You might ask, what is the Chief Operating Officer doing here? I'm a PhD, I'm a molecular geneticist by training. I actually started my career at Millennium Pharmaceuticals, my biotech career at Millennium Pharmaceuticals over 22 years ago, being a gene hunter, and it's really exciting, 22 years later, to now try to correct those same mutations that we were hunting back then. So what I'm going to talk about is tell you a little bit about how we're taking this fantastic technology and trying to develop therapeutics. And when you think about sort of medicine, where we started with small molecules, vaccines, antibodies, now we're using, and I'm going to steal a language from the CSO of Sangamo, it's using DNA as therapeutics. CRISPR allows us to do that. So what we're trying to do is really exploit, really exploit this technology to capture as many diseases as possible. And when we think about developing this technology, we have to think about it in two different areas. There we go. Oops. I was told that I had to. There. We have to think about the required performance. How efficient does it have to be? So how many cells need to be edited? And then what type of editing do we need? Will we do an ATJ, just make a cut and we anneal it? Or will we actually need to repair? Or if we have to repair, do we have to do it in an allele-specific way? Those are all the things that you have to take into consideration and also optimize around those issues. Then you have to think about the required selectivity, and you can do it around the specificity of the guides and the promoters, but also the delivery. So this is how we think about the technology. But as we think about developing and scaling this technology, granted, it's new, but after a year and a half of being a company, we want to take it to the next level. Where do we start? We optimize the guide RNA and its protein, and there's different species of Cas proteins with different recognition sites that allow us to, again, capture a broader set of the genome. Then we go and optimize, with the guide, then we optimize with the protein. Then you say, what delivery modality do we use? And then, this is not most, we spend more time clicking then. Then how do we regulate its expression? And then, most importantly, what type of repair? CRISPR-Cas9 is terribly exciting, but it's just an enzyme and a guide RNA. You think about the guide RNA as the software for the hardware. It helps you to get there. But the real ball game happens in how the cell repairs that lesion. And so how can we manipulate? It has nothing to do with CRISPR. There might be an effect how long the protein sits there, but it's really how can we manipulate the cells in a safe fashion to drive one type of correction versus the other. So we're spending a lot of time in this piece of the pie and a lot of investment, and actually for a company, a lot of basic research. Granted, the genome editing community and the repair community are coming together to understand how we exploit these pathways to develop therapeutics. So this is the core platform, what we have right now, what we license from our founders, and what is the state of the art. But in order to capture more of the genome, you have to expand and continue to optimize. So you think about the guide RNA, novel chemistries, novel structures, small or bigger to make them more specific. The Cas protein, different species, but also hybrid proteins that allow you to have different functionalities. Where do we deliver it? Right now, as Chad said, very right, much of the nucleic acid delivery field has been challenged with this, be it plasmids, be it siRNA. Delivery is one hurdle that we have to overcome. while we're not spending a lot of time, like Alnylam necessarily, ourselves developing novel delivery, we want to leverage what the gene therapy world has done and also invest in others. And the way that we think about this is, what are the products that we need to develop in what tissue, what cell type, and then where do we go and work with academics or industry to allow us to develop that, to advance that delivery modality. Expression is another thing. Think about novel promoters that can be specific for a specific type, specific tissue or cell type. Then how do we regulate this protein, especially the Cas protein? Do we want it around all the time? Are there molecular switches that we can have? The protein comes in, does its repair, and gets shut down. That's absolutely critical. In the literature, you've seen that when you think about the ratio, and if you think about it as kind of a therapeutic index, like a small molecule, efficacy versus tolerability, we want efficacy and we want specificity and activity versus off targets. And there's data out there that having a quick burst of activity of the protein, being the protein or the message when you deliver it to cell, while it decreases the activity, it also significantly decreases the off target. And again, the last quadrangle, the last piece of the pie is. Next time we'll bring up the The last one is really the whole DNA repair mechanism and how can you tweak against the cells. Now I'm going to switch in a little bit and talk about our products, because everything that we do is done in the context of a specific product. Well, we spend a lot of time on the technology. It's really the product pull that allows us to, that forces us to improve that technology. So before the company was launched in November of 2013, we have spent, I don't know, I would say at least for me, about 80% of my time talking with clinicians, with translation of people, and evaluating the 7,000 genetic diseases and saying, where do we start? We want to be very thoughtful of advancing this technology, not be ruthless and try to jump hoops, but where do you start where you can validate the technology in a specific, let's say, indication in a therapeutic area, and then that opens the whole space because you deliver to that target cell, you know something about delivering to that tissue, et cetera, et cetera. So we've prioritized those diseases around sort of near-term diseases and long-term diseases. Of course, we would only go where there is a medical need, right? You start there. If there's an antibody, if there's a small molecule, in this day, and at least in potentially my lifetime as an employee, I don't think we're going to go there if there's a small molecule. 20 years from now, 30 years from now, we might. There also has to be a clinical and regulatory path. There's incredibly interesting biology, let's say, around neurological diseases. Developing a clinical trial with not well-defined endpoints, with disease progression that is very long, is much more challenging when versus an endpoint that is very well defined, something that you can measure in the blood, let's say. And also the clinical aspect, how many patients, where many of us will be starting with rare diseases where there may be only 50, 100, 200 patients, and working with the right centers that have an understanding of natural history, that can work with us to be able to say, yes, we're seeing an effect, and that effect is significant. So those are important. And then delivery. We'll start where there are deliveries that have been validated in the clinic, not necessarily around efficacy, but that they go to that tissue and that cell type of interest, and then what can the technology do now, and what can the technology do in the future? The way that we are developing our early programs is in close collaboration with academics, those people that understand very well the disease and can help us translate this exciting technology and moving it into patients. And there's many projects that we will work on our own and take forward to a commercial stage. And there's other ones that we will collaborate also with industry because they may bring something to the table that helps us advance that technology. Right now, while I'm not going to give you a detail of all our projects, I can tell you that we're working on 9 different projects. Some more short-term, I say, some we can drive 100 miles an hour, only others we can drive 50, and others we can only drive 25 miles an hour. And the limitation are both delivery and the technology. Doing HDR, getting sufficient repair in certain cells to make a therapeutic effect is challenging. So we'll start with those where maybe you need less editing and maybe you can use an AGJ and when the delivery is validated. We'll push those faster, but we're starting now with even the difficult ones. So this is just a representation of how we develop our products. You start with a computational biology, design multiple, maybe 100 guide RNAs. You evaluate them in vivo in a very easily transfectable cell line, and there you look at both on target and off target. Then you take a subset of those, and you take the 2 primary fibroblasts that contain a mutation of interest. And again, we triage for those that have more activity. And once you have a subset of those, and I'll give you an example, you go to a product prototype, you put it in the delivery vector of interest, and again, you look at efficiency and specificity. Because both efficiency and specificity will vary from cell type because of the level of expression of your target interest, or let's say superposition of the DNA, or the genetic factors. There's different levers that we can move along the way. One is optimizing the guide RNA, the Cas protein, the species, the expression and regulation of either the guide RNA and or the protein, and then the delivery modality. Now I'm going to show you progress in 2 programs that we're working on. First, LCA10, and then there's some progress in both editing T cells and HSCs. So legal congenital amaurosis is a heterogeneous set of diseases that cause retinal degeneration. There's many different LCAs. We are focused on LCA10, not LCA2, which is ongoing. It is part of a program for a gene therapy company called Spark. And they're bringing in a gene called RP65. So LCA10 is heterogeneous, and we're focusing on a gene called SEP290 that makes up a majority of these LCAs. And here, it's a mutation that causes early onset blindness. The reason why we focused, and this is another way of triaging diseases that we're going to tackle. If it's easy to put it into a vector that you can deliver it with traditional gene therapy, and it requires enzyme replacement, that's not where we're going to start. You could envision in the future there being genes that indeed are small enough to put into gene therapy, but have some very, very tricky, or I can't find the word, but something that has to be very finely regulated. In that case, you do probably want to replace the corrected gene into its own genome regulated by its own promoter to maintain, as let's say, the cell differentiates. But for the moment, where we focused on are repairing genes that are too big to put in traditional gene therapy. So this gene is big, 7.5. While we could put it into lentivirus don't have great tropism for photoreceptor cells. Interestingly, this disease it decreases the amount of photoreceptors on the periphery of the retina, but not in the central part. There are still photoreceptors enough that they're still active so that you can repair those. And most importantly, the neuronal surface between the brain and the photoreceptors are intact. In many diseases for the eye, that's gone. So while the photoreceptors are dying. So the fact that up to, let's say, age 20, those neurons are still there makes it a very attractive target for gene editing. So what have we done here? The mutation for this gene, again, is a wonderful entry point for a novel technology. There's a mutation of a gene in the intron. It creates a novel splice donor site. And when in the wild type, an extra piece gets put of an exon, of exon 26, and then it creates a stop codon. So what we did is we tried both single and double guide RNAs within the intron to get rid of that piece, and it was more efficient to use 2 guide RNAs because of the sequence within, or there's an arrow repeat, et cetera, et cetera. So what we've done is use 2 guide RNAs and the Cas protein, delete that piece, and then we can have normal splicing. So here, what I'm showing you is our top 7 guide RNAs, pairs. I think it's 8 or 9 different guide RNAs that make up 7 pairs, and you can see that we are indeed deleting that intron. This was done by a droplet PCR. And then here, what you can see is the combination, the combination of these guide RNA pairs. And what I want you to pay attention is here, these 2 controls are no guide RNA, so you have a lot of mutant in those cells and very little wild type. And then you can see with various pairs, you increase the level of the blue, meaning you increase the level of the wild type gene. And when you sequence those cDNAs, we have shown that it is actually the wild type cDNA. So we're moving forward with this as a product. Now we've put them into different AAV vectors. We've shown that we can package them efficiently. We confirm that we have activity, that we delete what we had said we would do with 2 different promoters regulating that Cas protein. And we've sent these constructs to Bud Tucker at the University of Iowa. We have a close relationship with the University of Iowa. They have a fantastic center. They treat a lot of patients with genetic diseases of the eye, and they have about 9,500 different patients, and they've isolated the fibroblasts from these patients, and so you can do a lot of genetic analysis and kind of editing with them. So the idea is that they will, in their GMP manufacturing facility, make up the AAV vector. Interestingly enough, Bud works with, takes iPS cells, differentiates them into photoreceptor cells, and can look at activity. The defect, this is a structural protein, septinone is a structural protein involved in celiogenesis. And so there are celiogenesis assays that we're doing in-house just with our fibroblasts when we starve them, but he's doing them in these eye-cup models, so in an ex vivo model to look at activity. And once we see activity, and we can model based on the patients how much editing we need, we can go into non-clinical development and quickly into the clinic. So that's where this program is, and we have other programs in the ophthalmology space that we're moving forward on. Very quickly, I'm going to switch over and talk about blood. So while everybody's excited about editing blood cells, they're incredibly finicky to work with. And so we have spent a lot of time trying to edit both T cells and HSCs. And I'll show you some data very quickly. Here we're taking T cells, both immortalized cell lines K8562, DRPET cells that are more differentiated, and then CD34 positive cells. We see a good editing, and most importantly, these cells are viable. And this was done with RNA using electroporation. What we've also done is compared traditional electroporation, and here what we're electroporating is taking the messenger RNA, the Cas protein, and the guide RNAs, and then we compare it with a ribonucleotide complex having the protein and the guide RNA and then the lipid, and how does that compare? And they're both pretty efficient, and you get a higher percent of editing with these ribonucleotide complex, and again, the cells are viable. A lot of effort that was put in Chad has worked, and I think he knows the sweat that goes behind editing HSCs, and we spend a lot of time modifying both the protein and the guide RNA to get effective editing. And here we've edited CD34 HSC cells. They're viable, and most importantly, they maintain their stem cellness, so they differentiate into the right cells. So I'm going to skip quickly. We can also multiplex and edit different cells. And by the way, we're not working on CCR5 or PD1. These are just wonderful models because other people have worked with them. And ultimately, if we wanted to do it, you could say, okay, how does this editing compare to a Samuel-like product? But that's not where we're going. They're just wonderful models to work with. So we can multiplex. That's the other beauty. Oopsie daisy. Can you put it into slide mode here? Another advantage of CRISPR that you can think of, you could target multiple genes at the same time. Are we starting there? No, not yet, because you want to control your system early on. So here I just wrote down some lessons learned as a company developing products, developing novel medicines with a very new and exciting technology. When we started the labs in February of last year, this is the conclusions from where we are today. Guide RNA characterization is much more challenging than anticipated. And when you see a lot of publications in the literature, they're fantastic, but they're binary data. I just need 30 seconds. It's plus minus. When you want to develop a product, you really need to choose and optimize and reiterate. The guide RNA selection and optimization actually requires empirical work. You cannot predetermine computationally. Editing primary cells is much more challenging than editing immortalized cell lines, and I would say that at least 60% of the data out there is with immortalized cell lines. And the issue is both engineering the delivery or the repair pathways, and delivery optimization is critical, and it will change with cell line to cell line or tissue type. Protein engineering will advance us. Protein engineering and RNP delivery will allow rapid generation and evaluation of novel Cas variants, again with the goal of being to capture as much of the genome. Novel assays are critical along the way for quality control and biological assays to make sure that we're doing what we set out to do. The product development model that we put in place is us focusing on the technology and working very closely with academics or industry to help us advance things into the clinical. And the question of animal work for product development of edited medicines is still a question, because your product only hits the human gene. So no model is really going to tell you anything about specificity or tolerability of that product. And as soon as we get more packages of data around our most advanced products, our goal is to sit with the regulatory agent and come up with a path. The reality is that just like for gene therapy or enzyme replacement, every product will have a boutique-like regulatory development path. And with that, I'll say thank you and take questions.
Sorry to just beat the dead horse, but just to follow up what I asked Chad about CCR5 and maybe when you talk about your prioritization in the therapeutic area. I'm just wondering if you can share your answer as well. Is there a tipping point when you would go after some of these targets where we do have drugs potentially, new therapies? So I'm thinking CCR5, right? You're talking about curing versus someone who's on drugs for the rest of their lifetime.
Yes.
PCSK9, right?
Or PCSK9.
Genetics tells you that you want to hit a RA57.
PCSK9 is a wonderful example, and there's just literature with animals with PCSK9 and genome editing with Cas9. But there's two antibodies that look very, very promising.
Yeah, but I guess mine is there typically when we say you want to invest in that just one-time therapy where you potentially cure them. This is some of these on drugs or injections.
Yes, very much.
Rest of their lifetime. So I'm just curious, you know, you guys think about this all the time. Is there a tipping point where you say, okay, for some of these targets, it may be worth go back and find?
I think that's a big point is when you have delivered to that tissue type and shown safety and efficacy. But I wouldn't start, I don't think it's ethical to go after a new technology when there's therapies that are safe and are ready to be on the market pretty
So
soon.
I the same as these are the way you have to AE as well with the liver, so that's what I'm asking.
Yes. Yeah.
It's
Well,
safety is not, right? Safety is concerned with the vehicle, not the reagent, not
Yes, but you also have to think about the biology. Well, everything that we put in our body, I spent 7 years in the nanopharmaceutical space. You put anything IV, it goes to the liver. Be it a virus, be it a nanoparticle, be it any type of particle. The liver is your, I'm not an MD, so I can use this word, is your trash, right? That's where everything goes. The question is, how much do you have to edit? How much do you have to dose? A and B, you can only dose once. So if you need repeat dosing, if you could get a lot of editing with cells and assume that there's enough repair that you have to only dose once, you use A or B. If not, if you have to do repeat dosing, you probably go with a lipid nanoparticle. Right?
So I guess in those cases, sorry, I'm turning this over.
It's going to be a quick short
In those cases, the reason I ask that is, right, it doesn't work with first dose, and let's say everything's safe, you're not looking at something else, at least then you still have these doses that are available.
Yes, but I guess, like I said to you before, it's near term versus long term, and you have to de-risk certain things. And you want to go where there is unmet need. You want to go where doctors really, and I've sat down with many of those, they show us children, right, where there is no cure. There's muscle diseases, there are eye diseases. That's where you start, right, because there's nothing else available. And then you move on to a chronic illness like high cholesterol.
We have one very quick question. The same quick question is about off targets, because I suspect that you spend a lot of time characterizing, like you said, your eyes to make sure that they're very clean. Have you ever seen evidence of functional consequence of off targets when you detect if there is a cut? Have you ever seen, that actually there is a functional consequence in vitro and in vivo?
We have, because we don't test for function with the off-target. We haven't gone in vivo. We haven't made an animal model, but for ex vivo we look at it, and of course there may be something that grows. I mean, the off-target is something that everybody talks about, and it's very locus specific, right? So a priori, can I tell you that everything that we do is going to be absolutely exact? No, but that's why you iterate around the product. And each guide RNA may get a different off-target, so a priori you can't really determine. The off-target is not a generic answer. It's very dependent around the guide RNA that you're designing, and the Cas protein, and the tissue type, because it could be that a gene is very lowly expressed, and it hits that there is an off-target, but you get no cutting, let's say, and that's okay. Thank you.